Okay, let's get started. So this is an introductory presentation about single-cell RNA sequencing. This is a little bit different than all the other presentations we've done for the last almost 4 days. The technology is still improving. It's not like a whole genome sequencing, which has been around for more than 10 years. This is all very new. And everything changes every day. So you'll see my last slide, I have a whole list of tools, and by next year when I come back, they'll all be different. Okay, and I will say that. So just keep in mind that it's a very quick changing new field. And so we're going to give you an introduction to what's happening, to what single-cell sequencing is and what you need to do. And there's no command lines to run in with me. I'm not going to show you any GATK command lines. And in fact, we don't actually have GATK tools. There are people on my team who develop tools, and they're in the GATK family, but you'll see they're not actually GATK. And there are many people around the world who develop tools. In the early days of next generation sequencing, there were lots of tools and no one knew what to run. We're like that now with single cell. Now with bulk DNA and bulk RNA, the normal, you run GATK and you know what you get. It's easy. Okay, so I just want to give you the background there that this is very new, and I'm going to teach you more about the science today and what you do and when you see people giving you plots of the single cell sequencing, what that all means. Okay, and why it's so hard. But I'll motivate it a little bit with some of the background biology. So if you only study DNA, you learn a lot, but you don't learn everything. Okay, everyone assumes from biology class that you have DNA, the RNA, the proteins, and that's it. Simple. It's a nice linear progression, right? So if you've just done high school biology, that's what you think. But if you actually learn a little bit more, you learn that it's actually a lot more complicated than that. And it's not a linear process. Okay, first of all, the whole structure of a cell is not 2 dimensions. Okay, even though in IGV you look at DNA really 2 dimensions, really 1 dimension, just a string. It's not like that. The cell is 3-dimensional, and it's very complicated. And every protein interacts with another protein and sends a whole cascade of information. All that process is important to understand. And so understanding just how the DNA works is not enough to understand how a cell works. Okay, if you can imagine, you're trying to understand the workings of a cell. If the DNA is your code, your software engineering code, then the RNA and the proteins, let's say that's the output of your code. So we're trying to reverse engineer. We're trying to understand what's happening inside a cell. And so we have to work back. Really, the RNA helps us work back and understand all the complicated things that happen. So as an example, on the right, this is signal transduction pathway. This is how one protein affects another protein and interacts with another one. And there's a whole pathway of what happens. And understanding, and every cell is different. Your heart cells are different than your skin cells, which are different than your brain cells. And they're all using different proteins. The same genes, but not the same proteins. And so understanding how they all work is very important to tell us how cells work and how, therefore, the whole body is working. Okay, so it's not just enough to understand the genome. We have to understand the cells too. Okay, so why single cell resolution? Why do we do this? So just to be clear, biology is 3D. Right? It's not 2D. It's not 1D, and it's actually 4D. Right? What am I missing? Time. And time also comes into play as a cell. A cell is moving, and things are happening over time, and different genes are being expressed over time. And so all of that is super important. So there's some examples here, as you can see it. The genes being expressed inside a fetus change as the egg expands, as the embryo expands, and these cells start to differentiate in different parts of the body. Similarly, with a tumor, different genes are expressed in different parts of the tumor along the evolution of that tumor. Okay. And in disease states also, right? This is an example of a diseased heart. Okay, cells, they're all different. Understanding how that all happens is super important to the biology. I'm just trying to understand, to motivate you, why you should study cells and not just DNA. Okay. This is a great picture to understand this a little bit. So I give credit to this to Aviv Regev, who used to be one of the lead researchers at the Broad. She's left since then to Genentech, but she's still in our hearts. So this is her metaphor. So again, imagine your body is a fruit salad on the bottom right, okay, made up of lots of different types of cells. But when you take RNA from your blood, you just take a draw, you take all these cells, well, you're just taking all you get is it all mixed together. You get a fruit smoothie. So in the smoothie, yeah, for sure, you can tell, oh, there's definitely a banana in here, and I think there's some blueberries, but you don't know, you can't see the resolution between them. You just know that they're all mixed up in the bunch. So we study single cells to kind of see the differentiation, the differences between the different cell types. Good? Understand? Okay. No, so single cell omics has, really started 10 years ago. And in the early days, you could not get that much, you could not do it well, not at large scale. So Tang et al. Showed that you could pull out RNA, small quantity of RNA. That's important, by the way, small quantity of RNA from one cell. And if they sequenced it, they could actually figure out which genes were expressed. But it's only recently that you can do that industrially, commercially, at a large scale. And there are companies, so one of the most famous companies, one of them is 10x Genomics. 10x Genomics commercialized some of the processes that had been going on in the last decade. So for the most part, when we think about single cell sequencing, we think about 10x Genomics. I don't work for them. They're just another company. We do collaborate with them, but just, I'm not trying to promote them as a company. They're the most standard and one used for the most part. But really, for sure, you can see this is an example. The plot here is log scale, so you can see the growth of the number of cells being produced over time. So really, things are moving quickly. So from a year ago, 10 years ago, these products you could barely sequence a single cell. Now you can get tens of millions of cells sequenced in one shot. So this is real now. And with single cell sequencing, you can start to see plots like this. And I will explain to you how to make these plots as we go along. Okay, but at a high level, what you're seeing are examples of UMAP or t-SNE plots. Okay, so again, I'll explain to you what these are and what the meaning of them later. But for the most part, when you see the colors all keep, so usually in these plots, you group cells that are similar together. So when I say they're similar, it means they express similar sets of proteins. You have about 30,000 proteins in the human genome. And so they're all together in the graph if they express something similar. Now, on the graph on the left, this is showing you the progression of a zebrafish. So they've taken 6 sets of cells from a growing zebrafish embryo, and they colored it by the time they took it. So there were 6 of them, so there's 6 different colors. And you see, as the embryo starts to grow, the cells begin to differentiate. It's a pretty cool plot to see something like that. You can see how things differentiate and they become very specialized as they grow. And so you can only do that when you can see a single cell resolution of an organism. Now the plot on the right is just an adult mouse. The reason why you don't have the same tree structure is because this is not growing. It's all stable. It's all one mouse. And so the colors there show different, again, they're all grouped. The clusters are by cells that express similar genes. And so now they're colored by the type of cell they are. As you can say, well, I know that, B cells, immune cells, all kind of have this, I expect these genes to be turned on and these to be off. And I can see that that's what's happening in this cluster over here. Okay, so here's epithelial cells, for example, and you can, epidermal cells, you can very easily see that different cells cluster differently, different cell types. And that's pretty cool. And that's the kind of plot you can make with single cells. Now, if you asked a researcher 10 years ago how many types of cells there were in the human body, they would have said hundreds, thousands. Now we know that's not true, because even within the epithelial cells, there's many different types of differentiation, many different types. And we only know that by studying cells at a single cell resolution. So we've learned a lot of biology because of this. I also this isn't in my slides, but I did mention that with cancer, it's really hard to, because the tumor is so different, so heterogeneous, it's so hard. But you actually pull out single cells of cancer too, and can see then you don't have your different allele fractions too. So there's a lot of single cell sequencing with cancer too. Okay, so let's talk about how you do what the typical single cell RNA-seq experiment works. So first, you got to get a biological sample. I'm not going to kind of go through all the steps here. This is an example. We took some heart tissue, or some heart cells, and you do the stuff you do in library in preparing your samples. And I should say it's very important to understand that every type of cell is prepared differently. So skin cells obviously are extracted differently than heart cells, which are different than brain cells, which are different than muscle cells. So there's no one method to do it for any type of cell. And we're going to have to deal with that later on in the quality control. Okay, but the point is that once you're done, at the end of it all, you have a vial of cells in some sort of buffer. Okay, so I just have some cells. Great. Now, what we need to do is go from RNA to cDNA. That's how you can sequence it. That's with normal bulk RNA sequencing. They take the mRNAs and they turn them to cDNAs and they sequence them. Okay, so we're going to do the same thing here. The difference, though, is we want to make sure we do it so that each cell is different. That's the hard part. How do we turn the RNA from each cell into DNA, but keep them separate? So you can do it like the low throughput way on the left, which is pipetting them individually. I'll pipette this cell into this well and this one. That takes a long time. A lot of grad students. So, right? Or you can do microfluidics. And that's what I'll show you the 10x approach. Again, there are other approaches, but I'm going to teach you the 10x approach because we know it the best. All right, so this is actually, so because it's a PDF, this doesn't show animation, but this is actually pretty cool. The innovation here is that you can take a stream of oil, and you can push water into it. And when water and oil don't mix, so what happens is that the water turns into water beads, like circles of water. Yeah, and so they're all separate now, floating around, and they can push through the process. They try to push single cells, into each one of those water droplets and a bead, a specialized bead. So they push a bead and a single cell into each of those water droplets. That's what's happening there. Okay, and what's happening in that, what those beads have are chemicals that lyse the cell. That means it breaks apart the membranes of the cell, so the cell explodes out, and all the RNA is floating around. Okay, so one thing is they lyse the cell, and they add barcodes to it. Now each water droplet will get one bead. That bead has tons and tons of barcodes, but the barcode's all the same. Just like in the supermarket, all the same cans of toilet paper, it's tuna fish, toilet paper, they all have the same barcode, so you know the same brand, right? So in here, all the mRNAs are going to get the same barcodes for that cell. So they're all contained inside a droplet, and RNA from a different droplet get different barcodes. But within a droplet, they get the same barcode. And they also get something called a UMI. We'll talk about that a bit. Okay, and so the UMI we'll talk about in a bit. But the nice thing here is you can now tell which RNA come from the same droplet, from the same cell, because they all have the same barcode. Here's an example of what that looks like. There, you can see the cell inside a water droplet with your little bead. Pretty cool, right? Now I mentioned there's barcodes. They also add something called a UMI. Okay, The UMI is the Universal Molecular Identifier, I think it stands for. So the UMI is actually a randomized sequence. Now, unlike the barcode, the UMI is different for everything. They're all different. There are millions of different possibilities for the UMI. UMI is just a sequence of bases. And the reason why you do that is because we're going to have to remove duplicates. Just like we had marked duplicates in DNA, we're going to have to remove duplicates here. And the UMI helps you do that. So within a cell, all of the RNA will get the same barcode, and they should hopefully all get different UMIs. And then we do PCR later. If we see the same UMI, we know it's a duplicate because they all have different UMIs. That makes sense? They also get, so they get adapters for PCR, and they get a poly T. So our RNA has a poly A tail, so they get poly T tails, so they can hybridize. And I think that's all they get for that. But now, what you can do is you can take all those water droplets and break them apart. Now everything's floating in one big soup, but that's okay because they're all barcoded. So when I go sequence, I throw that all into a big test tube or whatever it is that, and you can put them in a flow cell. You can now sequence that data the same way you can sequence normal DNA and RNA because you've added specialized barcodes to it. That makes sense? So for the rest of the sequencing process is the same as you'd normally do it. Here's an example of what you'd see actually in a BAM file. What you basically see is that read one will give you the cellular and molecular identity. It'll give you the UMI and the barcode. It'll tell you which cell it comes from and whether there's any, whether it's a duplicate. And the read two, because again, you're paired in, will give you the actual sequence of the RNA, really the cDNA. Okay. You'll get about 100 to 500 million reads from about 5 to 20,000 cells. That's typically, if you do it well. Now notice that second bullet on the bottom, the RNA to cDNA conversion is only at 10% efficient. That means we don't always get the RNAs to turn into cDNAs. So we sample 10% of it, but it's not perfect. So that is probably one of the most inefficient pieces of the single cell process. But we still do get a good sampling across the RNAs. RNA is a little bit more unstable than DNA. Okay. Questions so far? Make sense? All right, cool. So let's talk about that UMI, make sure we understand it. So what I've shown you a made-up example on the bottom. We've talked about TP53 as a gene. So here, the cell barcodes, remember, those should be the same within the entire cell. They all get the same barcode. So here, notice that's the thing on the CB on the left. That's the green piece. So notice we have a whole bunch of reads. We get 7 reads that come out, okay, on the bottom right. All of them have the same cell barcode. They're all that light green. And then notice for TP53, I see 2 reads with this pink UMI and 1 with light blue. Well, that means, again, we've done PCR. Just like we do any sequencing, there's the PCR amplification step. So it looks like we've read off the same molecule twice, even though they're different lengths, because they have the same UMI. And that UMI was a random sequence. So the chances of RNA having the same exact barcode and the same UMI and actually being different molecules are very low. It's possible, but very low. So we assume they're not. And this is how we remove duplicates. Okay, we convert them into really one. We remove one of these. With EGFR, notice there are 4. There are actually 4 duplicates. So this is how we get accurate counts of how many RNAs there without being fooled by PCR amplification. Good, makes sense? Okay. So in general, I would say the 3 stages of RNA are really parallel to the 3 stages of DNA. So just to step back, in DNA, when we talk about primary analysis, that happens on the sequencing machine. It's called base calling, and not variant calling. Base calling is what happens on the machine, where they have we showed you the fluorescent lights with Illumina, for example, and Emma talked about how PacBio and Oxford Nanopore work. So that is called base calling, and that's the primary analysis, going from physical material to letters into a FASTQ. And secondary analysis is the GATK in DNA, and right? That's what we do. You go from a raw BAM to a set of variant calls. And tertiary analysis is the GWAS and gene burden tests and all the stuff you do downstream of that. Well, the same is true in single cell. You have primary analysis that happens on the machine. Secondary analysis is going from raw reads to a matrix, and we'll show you that matrix, which is cells in one dimension and the genes by the other. And then the tertiary analysis is making all those. There's a lot of QC and work you have to do, which we'll go over, and then making those awesome plots. So I just want to, I'm gonna go over that list in the tertiary analysis. I'm going to remind you a few times. Okay, so the normalization you see, so denoising is because the sequencing is inconsistent with cells. Some cells get sequenced at higher depths, but they should all be the same depths, right? We normalize. So feature selection is getting rid of boring genes. Dimensionality reduction is finding which genes are co-regulated, and so we only have to find one of them, right? Two genes, if they're both regulated together, they always express the same levels of the same proteins. And we don't have to study them both. We know we put them together. We have to deal with batch effects because, as I mentioned before, different cell types get extracted differently, and so they all look different. So there's clustering we talked about a bit in the plots, kind of showing which cells look similar. Differential expression analysis is showing you which types of cells express genes differently. And I'll show you these in the plots. And then the kind of like the trajectory reconstruction is that zebra-fish plot I showed you before. You can see how cells change over time. And so these are all the things you can do, and I'll go over examples of most of these visually soon. All right, so let's talk about secondary analysis. Okay, we talked a little bit about the cell barcode. CB is cell barcode, the UMI, that random identifier. So you have to first align the read to the reference. Nice and easy, right? We've done this before. You don't usually use BWA for this. You usually use an RNA aligner. I think someone talked about this before. Maybe it was the long reads flow. I don't remember when we talked about aligning to transcriptomes, but it's important here to notice that your RNA is going to be made up of DNA, right, comprised of the DNA from different exons. But when you go back to the reference genome, you have introns interspersed. So you're going to expect the RNA to align with major gaps, which represent the introns. And so there are specialized aligners that do this. There's a version of BWA that does that, but I think it's called Minimap 2 that does that. STAR is an aligner that does this. So there are, those are probably STAR is the one that's used the most. Minimap 2, I believe, is written by Hung Lee, who wrote BWA. Okay, so after you've aligned things, you group your reads by the barcodes. And now the barcodes, remember, anything with the same barcode is from the same original cell. And so here I'm representing that by the leftmost piece of it here, these colors on the left here. So all the purple come from the same cell. All the blue come from the same cell, just to show you that. And the UMIs, then within that, you group by UMIs. And the UMIs help you remove duplicates, as we have already discussed. So hopefully this looks like we've seen. And the most important thing is to output a cell by gene matrix. So usually this is 30,000 long on the x-axis because there are about 30,000 genes, and way more than 3 on the y-axis. You usually end up having many, tens of thousands or millions of cells sequenced. So this gets very big. In our example, it's small, so I can put it on the PDF, right? But usually this is a very, very big matrix. That being said, as you'll see soon, it's a sparse matrix because most of the cells will be 0. Okay, it'll be 0. Now here I didn't call it cell by gene, I called it cell by droplets. And we'll explain why, because I haven't kind of explained to you why those are zeros, okay? And the answer is that most droplets are actually imperfect. Technology is not improving, is improving and is not perfect yet. And most of the droplets end up having no RNA, no cell. Okay, so there would be a lot of zeros there. Okay, so also sequencing is not perfect. So I'm just going to show you some points on the bottom. Sequencing is not perfect. And there could actually be errors when you sequence the cell barcode. All right, so which means that even though like this, it's 10 bases, it could be only not matching 9 of them. So you have to do a little bit of correction to see which cell barcode is actually accurate there. And the same thing could be true with the UMI. There could be differences in the UMI too. And you have to correct for those just a little bit. So that's important. And I guess we'll go and talk a little bit about some of the tools I mentioned. So in creating that the alignments and the matrix, the most common tools are listed here. Cell Ranger is developed by 10x Genomics. I don't know what the license is, but it might not be fully open source. So just want to let you know that, mention it. Actually, the Broad itself has its own process for the secondary analysis called Optimus, but it just uses Star Solo, that middle one under the hood. So we just use Star Solo ourselves. Okay. So we've got this matrix from the previous page. We have that matrix, but we're not quite done with it. What do we do with it? We got to do a lot of processing. So technically, we're in the DNA world, this might fall into secondary analysis because there's a lot of processing we have to do to get the variants we're happy with. But its single cell is so new that this is now tertiary analysis downstream. Even some of the quality control is tertiary, is new and novel. So I'm going to kind of go through the steps here, how to go from a count matrix to something much better. So again, we have on the bottom left, imagine we have a cell matrix. So you have genes on one dimension and your droplets on the other. And so what you're doing is you're counting all the unique reads for each of those genes in each one of those. Just to make sure that makes sense, I'm going to go back a little bit. So for example, here, I know there are there's only one unique read for TP53 from this cell. So I put 1 in that matrix. And let's find one there. So the ApoE here is 2 unique ones, right? Because they have different UMIs. And so you're just putting counts into a matrix. That's what we're doing. We're just counting them. That's all you have to do. It's pretty simple that way. And you can see very soon that some of the genes that get expressed highly, those counts get pretty high. All right, so this is actually a typical kind of matrix you would see. So notice the second and fourth rows here have like counts of 0s, 1s, and 2s. Okay, so why does that happen? So one is that I mentioned not every droplet gets a cell. In fact, like 95% of droplets don't get cells. So this matrix is pretty bad. We only care about 5% of the rows. So you got to remove them. But notice the ones that don't get cells still have some counts. Why is that? And it turns out there's just ambient RNA floating around in the soup. Okay, when you break up cells, there's normal cellular RNA, and there's other random RNA that is part of it. And so it just to make our lives hard, we have to contend with that also. Okay, so it's very easy to go to this row and say, look, I can tell there's nothing here. But even some of the counts in a good row will have RNA that isn't actually from the cell, called ambient RNA. And so we got to model that. And that's what a lot of this quality control is. So the one easy way to figure out which rows are bad is to start just to rank them. Okay, count up all the total RNA, count up all the sum of all the unique reads in each of them, and plot them here. And again, this is just a toy example, but you'll notice if you plot them, there's usually a cliff. There's usually a very big difference between cells that have RNA and the beads that droplets that have RNA have a cell, and some that don't. It's usually very obvious. And so you find this cliff here, and that's where you cut and say anything above is good, anything below is bad. Great, right? Nice and easy. The problem is that it's never that easy, right? So so this is the tool that we've developed at the Broad. It's called Cellbender. So just as some background here, the GATK 4 had a nickname, a code name, which was Hellbender. Hellbender, turns out, is a massive salamander. Okay, it's actually a really ugly salamander, but the person who was leading development loved salamanders. So, and he thought Hellbender sounded like a cool name. So when we started developing single cell, they thought it'd be funny to call it Cellbender as that. So that's why they've called it this. Okay, so Cellbender does it does exactly that, but a lot smarter. It's more rigorous. It uses a generative model, so this is now machine learning. And instead of trying to find that cliff, it actually models the ambient RNA. So it goes back to the soup and tries to model how much it sees floating around there and use that to subtract things out. So I would highly recommend, if you're ever going to do single cell sequencing, use Cellbender for the QC. So Cellbender, really the GATK tools are used for the QC and preparation, not the downstream analysis. We get you ready to do your analysis. Okay, so this is a lot of kind of what I described before. I just want to point out that there are rules for QC, and none of the rules are they can all be broken at any one point with single cell, right? And again, because there are differences all the time, but the different cells they're all different. So I want to just show you as an example here a plot on total counts. Notice that there's a big cloud of where most cells lie. This is just their total counts of UMIs, of unique. And then there's a whole bunch that show up here. Sometimes 2 cells can sneak into one of those water molecules. And I have twice as much or even more than 2 cells. So you have to account for that. Obviously, that's not going to be accurate. So you need to account for that also in your quality control. And stuff that's very low, we want to account for. We talked about that. So none of this is super easy. One thing to mention also is that so mitochondria also express, and ribosomes express RNA2. And those, first of all, they're always expressed. So the genes, they are not interesting at all. And depending on the stress of the cell, they express higher or lower. Okay, so we definitely want to remove those too. So again, when you break apart cells and stuff, cells get stressed. Right? So things happen. So all this kind of stuff happens in every cell, and you have to deal with that in the QC. All right, there's a little bit of math here. Don't worry about it too much. When you want to look at things, you want to kind of look at them at the same scale. Now, sometimes when cells lyse, they don't lyse completely. So remember, lysing means breaking apart the cells. So they don't break apart completely. So you can't sequence all the RNA that's in that cell. So they get sequenced lower numbers. But it could be the same. It could be a healthy cell. You just don't get to see the numbers. So you don't want to get that confused by another cell that got full sequencing. And the sequencing depth is never the same between 2 different cells because sequencing is not perfect. Right? As you pull molecules out of your library, you don't always do that consistently. And certain genes will express really high numbers, and certain genes that are expressed will just express at lower numbers. But those are both turned on. So those you shouldn't have to discount one over the other. The ones that express at high levels shouldn't overpower your ability to analyze both of them. So there's a lot of normalization that happens here. And I don't necessarily want to go through. The most important piece at a high level is you first put things in log scale. That's important because therefore, when something is expressed at 10,000 reads, another is expressed at 100 reads, well, that's a lot in non-log scale, but on log scale, there's a difference of 2. Okay, so you can visualize that very easily. And then you turn it to z-scores, that bottom piece. That means you rescale things. So it goes from -1 to 1. So imagine your bell curve. So anything right in the middle of the curve is 0. Anything on the right tail is at 1. Anything on the left side of the tail is -1. Now everything is, all you're doing is normalizing here your numbers of those counts. That's what most people do here to visualize it easily and not have any one number dominate. So if something is on the right side of the tail, it's a 1. That's not a 10,000. And that's what that is. So, and again, there are tools and workflows for doing all this, and you'll soon see. So you don't actually have to implement the math yourself. Okay, so we talked about dimensionality reduction. I had mentioned that certain genes are expressed very hard, very high, but we finally got it down. We have a count matrix. It's all normalized. We're great, but there are still 30,000 genes. Right? That's a lot of genes, a lot. And remember, not all those genes are expressed. We have a lot of zeros in our matrix. Some of them are expressed but very low, so they're 1s and 2s. So we don't really care about those anymore. Some of them don't tell you a lot about your cell, like I mentioned mitochondria and ribosomal RNA. So we want to remove a lot of that stuff. So that's what this dimensionality reduction is. Because the dimension, again, that means we have 30,000 genes on one dimension. That's 30,000 is a lot. Let's remove it down. So if you remove all those things, and also if you know you're looking to study immune cells, well, then you can further it down to only B and T and other immune cells, right? You don't have to look at every single gene that's affected. But assuming you know no knowledge, let's remove all the uninteresting ones. You can still pull it down from like 500 to 5,000 genes. That's still a lot. That's still a lot. So then you can figure out that a lot of the genes are co-regulated. That means the same protein or some other gene regulates them both, or they are always in harmony. So when one is expressed, the other is also expressed. Okay, well, I don't need to plot both of them. I can just keep them in they're one entity in my graph. So I can remove it down to like 20 to 50. Okay, so 20 to 50 distinct entities. That's still a lot to visualize. Remember, everything happening here is in multi-dimensions. So 20 to 50 dimensions is still a lot to visualize. When I showed you VQSR, I had to put everything two-dimensional on these. So the last thing you do is you run a principal component analysis, and you're only going to display the highest scoring components, principal components. And that's what these things are. So UMAPs and T-SNEs are the most common ways of doing this. And all it is kind of moving down from your space of 50 to 500 components to a few that you can plot on a graph. Okay, now the way to think about this, again, I mentioned is cells that are similar, express the same genes at the same levels approximately, will cluster together. Okay, that's what kind of we're saying here. And that goes by their nearest neighbor. And so we can kind of show, so we know that there are groups there, and we can color them together if they all look the same. And that's what this is. That's what the clustering approaches are for single cells. That's what they all mean. All right, now differential expression analysis means let's look at the cells that are B cells, for example, and see which genes that are expressed with them differently than T cells. And this is how we learn what's happening inside the cell, which proteins are active, which genes are expressing. Yeah? Okay, so as an example, if you see, so cluster 0 there, you see that IL7R is expressed at high levels, and it's not expressed in other cells that you looked at. Well, we'll know from the literature that that is a classic profile for the CD4 plus T cells. Great. Now, how do I know that? How do I know that that's the kind of thing that those cells express? Google. Yeah. That's a very bad answer, obviously. Okay, Google is not how you do great science. Well, I mean, they would tell you that. But so in your hands-on exercise, you'll learn a little bit about how you might do this without Google. Okay. Hopefully that part works. All right, and then visualizations. There's so many cool things you can do with single cell, and it's pretty awesome. the colors are all useful. There's just so many cool things you can do. We showed you before the zebrafish embryonic progression. One, I want to take just some examples here. I want to take a minute just to explain to you what's happening on the bottom left, because I want to show it an example of cell bender. So the thing on the bottom left is a dot plot, and the way to think it. So just I want to notice that some of the circles are different sizes, and they're different intensities. They're all the same red, but some are more red and some are lighter red. So the way to think about dot plots is the size of the circle is how many cells in that cluster express a gene. So the size of that cluster, for example, here, like this is a big cluster, whereas this is maybe a smaller cluster. So these are all similar ways of viewing the same thing. Okay. So the size here is the size of the radius of the circle corresponds to the size of your cluster. The more cells expressing it, the bigger it is. And then the color tells you the magnitude of the expression. So if they express it really high levels, all those cells, it's really dark here. But if they express it, at low levels, then it's a lighter pink. That's the way to look at that. Okay, so now I want to show you, given that, let's go back and show you an example of what cell bender does. Okay, so just to remind you all, since it's new, cell bender's job is to remove both the cells that have the droplets that had no cells in them, but also, more importantly, to model and then remove the ambient RNA that's floating around in the soup. And it does that by modeling that soup and subtracting it out. So here's what happens when you see raw data, and we have that dot plot. Okay, and so you see there's a lot of things that look like they're expressing different genes. Okay? But after you apply the quality control from cell bender, a lot of that noise goes away. So you can just see. I'm going to go back and forth a few times, there and there and there and there. There's a lot of noise in that mix that goes away when you start modeling that ambient RNA. Again, ambient RNA is noise, and the noise shows up. And when you model the noise, you can easily remove the noise. And that's very important for understanding the differences. Otherwise, your T cells all look the same, but it turns out there's multiple different types of T cells that you can see only when you've removed that noise. Good? Okay, I'm almost done here. So we've gone through the standard set of quality control that you'd want to run for a single cell. Cell time assignment, again, you'll do in the hands-on. That's saying I can see what types of cell these are. That's a fun project, and there's a very cool thing happening at the Broad now, where we're creating a service for people to use to upload their RNA, the single cell RNA cells, and have us annotate them and give them the assignments, and then they can take them back. So you will, I hope, get to see a little bit of that in the hands-on. And then you can do really advanced things. I'm not going to go through all the details here, but there's just like an unending set of things you can do when you can see the resolution of a single cell, and you have thousands and tens of thousands and even millions of cells for an organism. So hopefully when we come back next year, we'll start showing you all these cool papers. The papers are coming out now. We'll start showing you a lot of cool papers. We have a limited amount of time, so I can't do too much now. But the field is growing and growing. So I mentioned this before. This is my last slide for you. Just for your reference, again, the slides are all available for you to see. If you wanted to do any of those QC steps, here are a bunch of tools, not everything, a bunch of tools that you could use to run them if you ever wanted to. Right? So you can make sure that these are downloaded to the high performance computing cluster if you wanted them. A lot of these are not written by the GATK team, and a lot of them are not even written by the Broad. They're just available. Like I said, if I come back next year, this list will be totally different because it's a growing field. But for now, this is what you use. Okay, and so with that, I believe that's my last slide. It is, yeah, for single cell. So I will take questions happily. I will say I'm not an absolute expert in single cell. I've read a lot of papers and I've been part of the process, but I've never actually processed single cell data myself. So I've learned a lot and been involved in papers, but as a PI and not as the one doing the work. So I'm happy to answer questions if I can, or I might just say, I don't know the answer, but I can tell you who would know. He's not here, but we can give you his cell number and you can call him. Questions? I'm just going to give it a minute also for folks next door if they want to come ask questions. And then I will ask Anton to come up soon. You'll have your last module, but you can't leave until you do the surveys. But the last module, which is the hands-on exercise for single cell. Again, this is all very new for you. For us, we're doing it for you. So again, just bear with us. I hope the module is good.